Good morning, students. Today we are going to start the crop improvement on kharif crops, and in this lecture we will be discussing about methods of breeding disease resistance. We have already covered introduction. We have already covered the losses that are or that were taking place as a result of origin of diseases. We often discussed the concept behind the development of diseases. We often talked about the mechanism that could lead or that could create a variety that is no more susceptible to the disease. And we were left with certain methods of breeding for disease resistance, right? So this is what we are going to cover in today's lecture. And I would like to tell you, like when we go for breeding for disease resistant, we have number of methodologies by which we can create a variety that is no more susceptible to the disease. Again and again, students, I will be using or I will be putting stress on two terms. One is susceptible, and another is resistant. And these two terms are in turn related to disease. Disease, everyone knows, it is any abnormality that is appearing within the plant, and the result of the disease is the attack of any microorganism. And since it is a part of living organism, it is a part of biotic factor. So this is said to be biotic stresses, right? Now see, this is the disease, and say this is the plant. This is the plant, right? If any disease is affecting the plant, if the or in turn I can say if any plant is being affected by the disease, we call such categories of plant as the susceptible ones. Susceptible means they are having a very that means they are having a very that means they are having a very that means they are having a very low immune system. Not immune system, I will say, I can call it as a defense mechanism. Defense mechanism, right? That means I have here susceptible means plant is very easily attacked, can be attacked by the disease. So such category of plants are called the susceptible. That means they have lower defense mechanism, and this defense mechanism in turn is required for the prevention of any disease or for the prevention from any disease, right? So susceptible plants are the plants which are easily attacked by the diseases, and as a result, because the defense mechanism is not too strong, so this is the result. There is always the origination of the diseases, and these plants are the ones which are most affected. When we talk for the breeding, our very prior objective of the breeding is to have a higher yield. But if the category of the plants are of type that they are being again and again affected by the diseases, then the motto or then the objective of obtaining the plants with a higher yield is not an easy task. So we need to improve the genetic content of these plants, keeping in view that they have a strong defense mechanism so that they are no more attacked by the diseases, or say as a result they could give you a prior objective of the breeding, that is the higher yield. So the category of the plants which are no more affected by the diseases, they are called as resistant varieties. So I'm talking about only two varieties. One are the susceptible, that is easily attacked, and another are the resistant, that means they are not attacked by the diseases. This can be there that even if they are attacked, then it is not going to affect. If this category is attacked, then it is not going to affect the yield or other desirable features of the plant, right? So this is what we need to discuss. We need to discuss if we have the susceptible plants, then how they can be converted or they can be modified into the resistant plants, right? So this is all about our today's lecture. That is, we are to go for methods that are helpful in breeding for disease resistant. And here are some of the methods, like we can go for the selection, we can go for the introduction, mutation, hybridization, Soma clonal variation, which is one of the part of say genetic engineering, that is the plant tissue culture. And then we can go for the genetic engineering, which is one of the part of biotechnology. You must be thinking now, ma'am, why you are discussing biotechnology or plant tissue culture over here? Because if you people remember, then we are to talk about the methods that is involving both the conventional techniques, and as well as it is talking about the techniques which are related to modern technology, right? So this is only the reason I have combined both the aspects within the methods of breeding for the disease resistance. So very first I will be talking about the selection. Selection, introduction, mutation, hybridization, Soma clonal variations, and genetic engineering. Very first is selection. Selection name itself is indicating that you are to select something, right? Selection means something is to be selected. Because you are talking for methods of breeding for disease resistance, obviously you need to select a variety that is resistance or that is resistant. You need to select a variety that is resistant. But the question is, very next question is, from where this selection could be continued. So when we talk for the very first methodology, that is the selection, then in this case, a selection can be done from the selection can be done from a population where a variety is commercially cultivated. So in this case, when we are going for the breeding for disease resistant, we will be selecting a resistant variety from the commercial variety. Commercial variety means the varieties which are already being multiplied at a larger scale. From those population, we will be selecting a variety that is resistant to the resistant to the disease, right? Why we are going for choosing a resistant variety from a commercial variety? The reason is very simple, because it is the cheapest and quickest method of developing a resistant variety. Because see, one will be producing any variety at the commercial scale, because it might be consisting of certain desirable features, right? Because if any variety is lacking the desirable agronomic features, then why we need to look after its multiplication? We are looking after the multiplication of the variety just because of the reason, because it is having the desirable agronomical features, right? And since we are already much familiar with the features, so it is easy to select a variety from here, and it is easy to select a variety from here, and it is easy, like we can go for the commercial multiplication. Now the next is, this is the commercial multiplication, right? Advantage is, like, this method has been utilized. This method is already utilized in many cases in the past. But the limitation is, like, see, we know that the commercial variety is the one variety that is already being utilized, right? But it has a limited usefulness at the present level of crop improvement, because picking up a commercial variety and selecting it for a disease resistant, nowadays it is not producing as much as good result as it was producing in the past. The reason may be Reason can be any. It may be environmental interactions, it may be some changes in the genetic content, anything can be the reason. Although it is useful in many cases, but it is not useful in all the cases. That means in every cases when you are to go for the selection, you cannot just depend on the commercial variety selection and to utilize this variety for the as a resistant variety, right? So this is all about selection. Name itself is indicating you are to select a disease resistant variety, and that particular variety is to be selected from a commercial variety, right? This is the concept behind selection. Now we will talk for the next methodology, that is introduction. Plant introduction, as the name itself is indicating, that means introduction. Something is being introduced, but that something is what? In the introduction, we basically talk about the plant introduction. We basically talk for plant introduction. So when we talk for the introduction, we have one area, and then we have this is the natural habitat, we can call it as. This is the natural habitat, and on the another aspect, this is the newer area. This is new area. And introduction is what? Introduction is picking up a variety from its natural habitat and then introducing it to the newer area. This is said to be introduction, right? But here, what is to be selected from its natural habitat and what is to be transferred into the newer habitat? Because we are not to step back from our today's topic, that is all about methods of breeding for disease resistance. That means we are here talking about the creation of the variety that is no more affected by the diseases. That is, we are all working on the creation of a disease resistant variety. So here, from the natural habitat, a resistant variety is to be picked up. This resistant variety is to be picked up, and in turn it is to be transferred into a newer area. What is the advantage behind going for the introduction? This is a very simple, and because we are much familiar with the many resistant varieties, so it is a very simple and quick method of obtaining resistant varieties. And it has also served the purpose of disease control, right? But this method is having many limitations also. Limitations means this, as I already said, this is the natural habitat of the variety. But whenever this variety will be introduced to the newer variety, in the newer variety, because there might be another varieties also. So in the newer area, there is for sure the presence of many other diseases, right? There may be the presence of many other diseases. So whenever this introduced variety will be transferred, whenever this resistant variety will be transferred to the newer area, it may not, very first thing is it may not perform well in that newer area. Secondly, it may become susceptible to the diseases which are prevailing in this new environment. And it is not always like concerned diseases are only present here. Diseases that are related to the other races, right? They may also be present here. So these three are the limitations when we go for the transfer of the resistant variety from its natural habitat to the newer habitat. Very first, as it was giving performance in its natural habitat, it may not perform up to that level in the newer area. Then here may be the presence of diseases that are concerned to certain plant species, and it may be affected by those diseases, and it can also be affected by the diseases that are the result of other races, right? So this is all about introduction, where we are looking after the creation of resistant varieties by introducing it from its own area to the newer area, but transferring the variety to the newer area itself is having many problems. Next is mutation. Mutation name itself is indicating in the mutation. Susceptible variety this is susceptible variety, and this susceptible variety is converted into a resistant variety. Susceptible variety is converted into resistant variety by introducing, by allowing the exposure of this susceptible variety to the chemicals or say to the physical mutagens, and this process is said to be mutation. For example, resistance to Victoria blight in oat. That means in the oat, what disease was being found in the oats, we had Victoria blight disease. And oat was made resist to the bacterial blight disease by exposing the oats to the X-rays or to the thermal neutrons, right? So in mutation, what is to be done in mutation? Any variety that is susceptible to the disease is to be exposed to the mutations, and those mutations can be the result of those mutations are the result of those mutations are the result of exposing the susceptible variety to the X-rays or to any other source of mutagen. So basically, mutation can be done by the two concepts. Like very first is either it is spontaneous, that is taking place naturally, or it can be induced also. Some other examples where mutations have been done are like in wheat. In wheat mostly striped rust and burnt is found, and these are some are the examples where, as a result of mutation, disease-resistant varieties have been created. Then crown rust in oats, mildew in barley, rust in linseed. So these all are the examples where, as a result of mutation, susceptible varieties they are converted into susceptible varieties are converted into disease-resistant varieties. The next method we will talk about hybridization. And whenever I say hybridization, what should quickly strike into mind is where one variety is crossed with the another variety, right? And you know, when we are crossing one variety with another variety, we know these are the diploids. So here there is say there is a presence of two alternative form of the genes, and here see there is the presence of these two alternative forms of the gene, and whenever the next progeny is produced, some of the characters from one parent and some of the characters from the another parent, they come in the front and they combine together and resulting into the formation of a hybrid. This is what basically we know about the hybridization, and it is a very common method of breeding for disease resistant, because here the one because we know like the genes are consisting of alleles, and alleles can be either dominant or alleles can be recessive. Alleles can be dominant or alleles can be recessive. So always the expression is given by the dominant gene. Dominant is the one that is represented by capital letter, and this is represented by small letter. So this is the basic concept of hybridization before I go into deeper hybridization, right? Advantage is what? It is actually, this hybridization is actually serving two purposes. This is the basic concept. I hope it is clear to all. Now I will be hybridization here is done by two methodologies. Very first one involves backcross method, and the second one is involving pedigree method. Hybridization here is being done by two methods. That is, very first one is said to be backcross and other is said to be pedigree. When we talk for the hybridization is actually serving two purposes. Hybridization is serving two purposes. Very first is the case where we are crossing the two varieties, say variety 1, it is being crossed with variety 2. Here these two are the parents, right? And say one parent is very important, it is very first parent is disease resistance, and but along with the disease resistance, it is agronomically undesirable. That is agricultural point, with the agricultural point of view, it is not much profitable or beneficial. Agronomically it is undesirable, right? But we are talking for the another variety. This is disease resistant, note it carefully, but agronomically undesirable, and it is crossed with another variety, but the another variety is susceptible. That is, it is easily attacked by the, say, it is easily attacked by what we call disease, but this is agronomically desirable. This is agronomically desirable variety. So here also we are going to have a hybrid, and more we will discuss it in the backrows methodology only. So right now you just remember a very one chief purpose of it is to transfer a gene. So this is disease resistant gene. This is disease resistant gene that is actually required. But this variety is agronomically undesired. So what we can do? We can transfer this gene that is not allowing the attack of the disease. It can be transferred to the one that is susceptible to the disease, because it is agronomically desirable also. So what we can do? We can look after the variety that is having the characteristics of, that is having the features of resistivity, and look going after the selection of a particular variety that is resistant to the disease. That particular character can be transferred from that particular parent to the another parent. As a result, whatever hybrid we are going to have, that is going to be resistance to the that is going to be resistant to the disease, and also it is going to be agronomically desirable, right? But this is not as easy as I am just illustrating here. Hybrid is to be crossed in back cross. Like what we do, it is normally we cross it with another parent, right? What normally we do, we cross it with another parent, say parent 3. But here we need not to look after the newer parent. Here, here it will be crossed back to the parent that is giving disease resistant feature, because it is back crossed. Back crossed means we are not going in the forward direction, we are going in the backward direction. So this is the reason it is said to be back cross method. And back cross method purpose is like it is helping in the transfer of disease resistant gene from an undesirable variety to the desirable variety. So this is said to be back cross method. This is one of the objective of hybridization. Now in the second case, what we are doing, that is second case is all about, say, second case is where what we are doing, again, we are having the two varieties in the pedigree. In the pedigree, again, we have two varieties. This is variety 1. It is to be crossed with variety 2. This is very basic concept, and by closing your eyes, you can select one parent that is to be crossed with the other parent, right? But here what is done? Here disease resistant features, the very important thing is you need to select the parents on the basis of features required. Here disease resistance and some desirable characters of one variety, like disease resistant feature, so we have to select only, no, because we are breeding for disease resistance only. So disease resistant variety is to be selected for 100%, and this in turn is now combined with another variety that is giving certain, or say that is giving some other, that is giving some other desirable character, that is giving some other desirable character, right? Now we have combined here disease resistant variety and some other desirable characters of one variety, but the other variety that is selected, it is having superior characteristics, superior characteristics of another variety. Earlier what we said, it was disease resistant but was not agronomically desirable. That means if it is having desirable characters, that is, it is agronomically desirable. And this is the superior characteristic variety, so it is going to be super and duper agronomically desirable, right? So these two varieties are crossed, and when we are going to have hybrid, this method is actually said to be pedigree method. We will learn about it in detail in the next slide. So this is the basic concept. This is the basic concept of hybridization, and it is one of the most common method of breeding that is serving two main purposes. Very first is transfer of the disease resistance from an agronomically undesirable variety to the susceptible variety, but the susceptible is otherwise agronomically desirable. And the second concept of the second chief purpose of the hybridization is to combine. Here what we are doing, we are actually combining features of it, and we are combining the features of it, because as I already said, half of the characteristics are going to come from one parent and half of the characteristics are going to come from another parent. So as a result, it will be having combinations of characteristics of both the parents. So obviously, we are going to combine disease resistant and some other desirable characters of one variety, and it is going to be combined with the superior characteristics of another variety, what it is possible by a methodology that is said to be pedigree method. So very first we will talk about backcross method. Very first we will talk about the backcross method. In the backcross method, we will discuss some of its features that will help you to easily understand concept of backcross technology. This is very clear to all, like this method is useful in transferring gene for resistance. That is, it is useful. See this flowchart that I prepared, cross that I have made here. It is helpful in transferring gene for resistance, from a variety that is agronomically undesirable to a susceptible variety that is agronomical desirable and widely adapted, right? But have you decided, do you know, like backrows is to be carried with what parent? Like it is to be carried with, obviously it is to be carried with a disease resistant parent only. But you know, whenever we talk for the any gene, say disease resistant gene, if we are talking about, say it is abbreviated by DD, right? By D. And you know, this is the D is the gene that is responsible for disease resistant characteristic. But you know this also, like any gene is the combination of alternative forms of alleles. That is, within the gene, there is the presence of one dominant, this is capital letter, so this is said to be dominant gene, and this is a smaller letter, this is said to be the recessive gene. Now you are to cross, you are to perform back cross. This is sure you are not to cross it with the next parent, you are to cross it with back parent only. And back parent is also clear, like we are to proceed with the disease resistant parent only, that is also clear. But this backcross is what kind of backcross method is going to take place. This in turn depends upon whether the allele, now it can be no within this dominant gene, it may be like this allele is responsible for disease resistant, or it may be like this smaller gene is responsible for the disease resistant. So if this capital gene is responsible for the disease resistant, then it is then you have to prepare a backcross where the hybrid is to be crossed with the dominant gene. But if this is the reason for the production of resistant feature, then hybrid is to be crossed with this parent. I hope it is being clear to you all. That means what backcross program is to be adapted. This in turn differ because backcross methods are based upon like whether the dominant gene is being transferred or whether the recessive gene is to be transferred from agronomically undesirable variety to to susceptible desirable variety. This must you have read in your fourth semester also while you were covering the methods of handling for the methods of handling segregation generation. At that time also teacher would have told you the basic concept of backcross method, right? So here backcross method differ depending upon whether the allele that is giving the resistant feature is recessive or dominant. Very important. Recessive or dominant to that for susceptible. That means susceptible means it is always going to be, if we say susceptible, then we will always discuss it. It is the smallest. Okay. Now the disease that is giving the disease resistant in relation to this one, we have to compare with this one whether the gene is dominant over it or the gene is recessive over it. So the backcross method is going to be dominant backcross method or it is going to be the recessive method, right? But a very rigid selection is to be done for disease resistant, and the plant type of recurrent parent is also selected for. The recurrent parent means basically, see, there are two parents. One is this parent and other is this parent. The parent that is actually giving its disease resistant gene, it is said to be donor, and the one that is accepting the one that is accepting the disease resistant gene, that is said to be recurrent parent. That means again and again this parent is to be crossed with this parent, so that when number of these crosses will be made, at the end we will be having a progeny that is totally where this susceptible gene is totally replaced by this disease resistant gene, right? You must be thinking now, and this question must be coming into the mind of many students, like, ma'am, how many backcross methods will be sufficient for converting this susceptible gene to the disease resistant gene? Then I let you know, like 4 to 6 backcrosses are sufficient. 4 to 6 crosses are sufficient when you are to go for a backcross method. But if you combine this backcross technology, with an marker assisted selection, this I'll let you know later on. But here some brief I will give you. Like here, the variety is further combined with certain markers, and these markers gives genotypic information. This gives genotypic information. So whenever any marker is attached to any character, it becomes very easy to recognize that particular character, right? I say you all, when you all are in the uniform, it is very easy to recognize you are the students of which college, right? Because the uniform is varying from the, and the moreover, if I don't talk for the uniform, and if I say you wear your ID cards, that very clearly predicts that you belong to which class, to which college, and to which semester, right? That is said to be that in turn we can think about marker assisted selection. Basically, 4 to 6 back crosses are to be done, and at the end, like when say this one back cross is carried, just a minute. Say when this back cross is carried, say where one variety features are now known to you all, it is crossed with and one hybrid is produced. This will be again crossed with one of the parent that is giving the resistant characters, a hybrid 2 will be produced. This in turn will be again back crossed with the same parent, then hybrid 3 is going to be produced. So such kind of 4 crosses are sufficient enough to replace the susceptible gene with the resistant one. And at the end of, when at the end we are going to have a superior variety, that will be that, then we need not to cross it with the back parent. We can self among these two can be self, and then as a result of self, whatever progeny we will be having, out of that progeny resistant plants will be selected, right? So this is about back cross methodology. This is about back cross methodology and how it is to be done, how it can be more conveniently done, like by using the marker assisted selection. And at the end, what is to be done? Progenies are to be self, means they are to be crossed among each other. And after crossing, the plants which are resistant to a particular disease, they are to be selected. So this is one chief purpose of hybridization, where the resistant gene is being transferred. Again, I repeat, this is very first chief purpose of the say breeding of hybridization, where resistant gene from one parent that is agronomically indesirable is transferred to the another parent that is agronomically desirable, but it is susceptible to the disease, right? So with this, I end up with the backcross method. So here are some of the more features that are related to the backcross method, like whatever progenies, here one more thing can be done. Now when one parent is going to be crossed with another parent, where we are talking for like those parents are resistant and they are being self. So we can select many such resistant homozygous plants, and in bulk we can go for the production of new resistant variety. And very important, whatever parent here, whatever result here, very important, whatever result here we are going to achieve, this is the one cross, but when 3 to 4 back crosses will be done, and when we are going to have a progeny, then it is very important to know that this progeny is similar to that is recurrent parent. It will be similar to the recurrent parent. And what is the recurrent parent? The parent where susceptible gene was present. Gene susceptible to disease was present. This parent is going to be similar to recurrent parent, but the very major difference is going to be this recurrent parent was susceptible to the attack of diseases, but this is no more going to be susceptible to the attack of the diseases, because within this recurrent parent, the susceptible gene is totally replaced by the disease resistant gene by crossing this parent again and again with the disease resistant parent, right? So the new variety produced is going to be identical to the recurrent parent, but except for the disease resistant. Therefore, yield trials are usually not required, and it can are not required for its release, because whenever we release a variety to the farmers, we basically go for we basically go for yield trials. But here, since we already know like the susceptible characteristic is replaced by the disease characteristic, so we know not to go for the yield trials before the release of the variety, right? This is about backcross method. Now we are to talk about the another method that is said to be pedigree method. So here there is no need to discuss now backcross method. We are only to discuss the pedigree method. So pedigree method. Concept behind pedigree I have already told to everyone. That means here two varieties, that means two parents are selected, and the two parents are like one is disease resistant, one parent is disease resistant, and along with the resistant feature, it is having certain desirable characteristics also. and those desirable characteristics are of one variety. Important thing. And with the parent it is being crossed is having superior characteristics of another variety. That means here we cannot say either this variety is agronomically undesirable and this is desirable. There is no certain differences among the two varieties. Instead, both the two varieties are having certain desirable and superior features, and moreover it is having disease resistant feature also, which is all about today's lecture, that is breeding for disease resistant. But before I actually tell you all before I make you all familiar with these breeding method, I would like to make you familiar with a certain terms. Very first is horizontal diseases. Here we will talk pedigree method in relation to horizontal and polygenic resistance, but one must know what actually is meant by horizontal resistant. So when we say for the horizontal resistant, here resistant means defense mechanism, right? This I told you in the very beginning only. Resistance is defense mechanism. Here plants have defense mechanism, and defense means defense or resistant. This is one and the same thing, right? Here the plant is having the resistant mechanism, and that resistant, very important, that resistant mechanism act. This resistant mechanism is acting against, one is either it is acting against single pathogen genotype, like it is acting against the genotype of single pathogen only, or either this is only, that means this is acting against one concept is only there, that is here plants have resistant mechanism, that is they have the defense mechanism. At that defense mechanism, very important, that defense mechanism is active against genotype of single pathogen. That means if here there comes many more pathogen, then it will not be able to resist from the attack of these pathogens, because it could defend itself against the genotype of single pathogen only. And pathogen is what is producing disease is said to be a pathogen, right? This is horizontal. That means direct relation. Horizontal means direct relation. It is producing, no doubt, it is producing the defense mechanism, but that defense mechanism is effective against a single pathogen, a species or genotype only. This is horizontal. This is horizontal resistance, right? Second, I will talk for the polygenic. Polygenic name itself is indicating, that is poly means many. That means in polygenic resistance, poly means many, that means here plants operate through many mechanisms. Their plant was operating by only one mechanism, and that one mechanism was what? That one mechanism was like it is controlling or it is active against single genotype only. But here it is acting by many mechanisms. And these, very important, these mechanisms are not concerned with the disease. Initially they were concerned with the disease, but now they are no more concerned with the disease, right? This is about polygenic. Like here, other disease control measures, like here fungicide can also be used. Like here, number of plant growth regulators can be used for controlling the disease. This is the difference among horizontal and polygenic resistance. So this method is quite suited for breeding for horizontal or polygenic resistance. Here, but what is to be done, because the disease is not always present, and when you are at the end, when you are to see the breeders that you have produced, like the varieties which you have created, which are disease resistant from your point of view, but you actually need to conduct certain trials to ensure that exactly the varieties which you have created are the ones which are resistant to disease. So here, for testing the varieties, like for conducting the yield trials, one need to artificially introduce the diseases. Like, see, if I say I have created this variety, and from my point of view, because I very perfectly followed all the procedures of the breeding, so with my point of view, this variety is for sure going to be the variety that is disease resistant. But when I say for the pedigree methodology, to ensure like really it is disease resistant or not, then we are to create in this environment artificially, we have to introduce disease into this environment. If plants could survive against this introduced disease that is artificially introduced into the environment, then we say we got the success. And if it is not able to if it is not able to withstand the attack of the disease, then it is not the one that was actually to be prepared, right? So this is the reason here artificially disease epidemics are to be generally created, so that effective selection could be done. And these all are the varieties which are result as a result of pedigree method. So this was all about hybridization. Now we are to talk for the next method, that is Soma clonal variation. This Soma clonal variation is also the modern aspect, and it is one of the part of plant tissue culture. But before I actually go into its two ways, like how disease resistant somaclonal variants can be produced, I will like to introduce you all with plant tissue culture, that is abbreviated as PTC, and its full form is plant tissue culture. So here what is done, plant tissue, that means plant tissue is taken and it is cultured. Say tissue can be any part. Say here I have selected the leaf. Part of it will be taken. It will be cut and it will be isolated. And on the other hand, one media is to be prepared that contains all the essential nutrients. That contains all the essential nutrients which are actually required for the growth of this tissue. And later on, this tissue will be transferred to this on the above surface of this media. Now, when this tissue will grow, it will consume the nutrients, tissue will consume the nutrients from this media and it will start growing. Certain plant growth regulators are also added into this media. And one of the plant regulator when added, this media is to be changed from time to time, because as the tissue will keep on growing, it will be utilizing all the all the nutrients which are present in the media, and very immediately, very sooner will come the condition when this media is deprived of any more nutrients. So this media is to be replaced with a newer media, and plant growth regulators are to be added into the end stages, at the end stages. Then from this tissue, there will be the development of root, and there will be the development of shoot, and the shoot will appear, shoot will bear leaves, right? This is the development of plant under artificial conditions, because these artificial conditions are being provided in the lab, and this is said to be plant tissue culture. But what happens while we are developing the plant from these under these artificial conditions, certain variations, certain variations they takes place, and uh, when these variations are taking place, these variations are taking place at the time of growth of the plant from a small part of the tissue, and these are only the variations which are said to be Soma clonal variations. These are the variations which are said to be Soma clonal variations, right? This is Soma clonal variation and the basic concept of plant tissue culture. Now I will enter into the Soma clonal variations. And when I talk for its features, then here, what kind of Soma clonal variants we need, which are no more susceptible to the attack of disease. That means here we require disease resistant Soma clonal variants. Variants means the plants which are developed after certain variations, they are said to be Soma clones. And here what we require, disease resistant Soma clonal variants. There are two ways of obtaining. Okay, you grown a tissue, certain variations took place while you were going through the following of the various steps of this procedure, and somehow variations took place, right? But you are to screen. Very first step is you are to screen here, and then you are to select the cell. What is done in the screening? These plants which are regenerated, this is said to be cultured, because culture is what nourishing the tissue through artificial media. Normally plant is acquiring all the nutrients from the soil, that is natural concept, but here it is receiving all the nutrients, it is nourishing itself by obtaining the nutrients say from nutrients from media. So this is the reason this is said to be cultured cell. The cell that is placed onto this media is called as a cultured cell. Cultured cell. Progeny means the generation. So here, very first, this is what I talked about, one plant regeneration. Regeneration means again it is generated from the tissue. When the plant is again generated from the tissue, it is said to be regeneration. Very first part is, this I talked for one plant. In the similar manner, number of plants are regenerated as a result of plant tissue culture, and in many variations must have took place. So here the plants are to be selected from these cultured cells, and very important, they are to be subjected to disease test. Now you must be thinking, ma'am, how this disease test is to be conducted? Then I let you know. When we talk for the disease test, what is done here? Here again the media is prepared. Suppose you know this is a variety A that is affected by a disease, say X disease, right? So what you will do, you will add here, disease is to be selected, so you will add here antibody. Antibody. You can say it antigen, any foreign microorganism is added here. If it is exactly consisting of disease resistant gene, this is actually the antigen that will produce a particular kind of disease, right? Now we place this plant here in this media that is having the antigen that is producing this disease, due to which this variety is affected, and this variety is also variety A. Right, we now need to see this variety A is having this disease resistant gene or not, and the resistivity is against what kind of antigen? X. So this is the X antigen that is introduced artificially like janbooj ke, we are introducing this antigen into the media, right? This antigen is X. Now we need to say whether it is affected by the presence of this antigen or not. This is said to be disease test. If it is not affected, that means it is having a disease resistant gene. But if it is affected, that means it is not having disease resistant gene. So this is the screening. That means here the plants which are regenerated as a result of plant tissue culture, they are subjected to disease test, and the ones which will grow even in the presence of this antigen, they will be called as a resistant plants, and those plants are to be selected, they are to be isolated from susceptible plants. Now very first is you developed the variations developed, and here see, you need to introduce, because it is from tissue, it is growing, so along with the plant growth regulators, you also need to incorporate the gene that is actually, that will actually provide it a resistance against this antigen, this disease, right? Now whether plant has incorporated that gene into it or not, this in turn will be selected by, this in turn will be ensured by screening methodology, right? Now what is to be next step is cell selection. Now cultured cells are selected for this resistant to toxin, because this is antigen, it will produce a toxin. If plants are resistant to this toxin, they will grow, otherwise they will not grow. In this way we can select for resistant to toxin, right? And then further from this plant, because then this plant can be taken out, and again from its leaf tissue can be taken, and in this way from this resistant plants many more plants can be developed, right? But you know, this is not most likely to be successful in cases where toxin is involved in the disease development. So I mean to say, where we have toxin, like we said, no, tests are to be, selection is to be done on the basis of toxin. But where toxin itself is responsible for the development of disease, there we cannot go for the Soma clonal variations. So this is all about Soma clonal variations. The next is genetic engineering. In genetic engineering, what is to be done? Genetic engineering means the desired gene is to be isolated, and it is to be transferred into your crop or into the variety that is susceptible. This is genetic engineering. That means here the desired gene that is giving the resistivity character, it is to be selected, it is to be isolated, then its multiple number of copies are to be prepared, and then it is to be transferred into the crops which are actually susceptible to this particular disease. So this have been several transgenes have been evaluated, like virus coat, protein gene, DNA copy of satellite RNA, defective viral genome, antisense construct. So these are the certain say genes. Gene. Now the gene that is being transferred, it will be called as transgene, right? So these transgenes have been evaluated, and out of which this one is the most successful one. So in the genetic engineering, when we talk for the bacterial and fungal pathogen, then resistant has been sought from the expression of these like transgenes. They, when expressed in the particular plants, they gave those plants resistance against a number of attacks that are conducted by bacterial and fungal pathogens. Like when genes in we actually selected the genes that encoded in sensitive target enzymes. So these all are the transgenes which when incorporated into the crops, they gave the crops a resistant feature against the attack of number of bacterial and fungal pathogens. These all are the transgenes only, right? The 1 to 5 transgenes, they are from they are taken from non-plant sources, and 6 to 10 involve the genes that are isolated from the plants, right? And whatever these transgenes methodologies are adopted, these all have showed increased resistance to the concerned diseases. But here in the genetic engineering, okay, we got one concept like we are to introduce a gene that will confer resistance to the plants. But you know what? Here, okay, you are introducing the genes into the plants, and that genes in turn will you have transgenes which are specific for concerned diseases, right? You have transgene, this is clear itself. But when we talk for the genetic engineering, like there must be taking cell death as a result of infection, right? What is done here, this is the main concept that will be discussed in the genetic engineering, that is all about cell death. Here we are inducing the cell death artificially, because if the cell will die, there will be no more generation of the no more copies of the say particular plant, say particular antigen that is actually responsible for giving rise to disease. So the strategy, very important, listen it carefully, that is the strategy of artificial programmed cell death. That means how you have to genetically engineer the cell. You will do certain modifications into the genotype of the cell in a way that the cell that is affected by the disease and the cell in which disease cannot be anymore controlled, that will be leader to artificially cell death. Because it may be like disease is growing slowly and slowly, and otherwise it may take 5 to 6 years to die, but we will program the cell in a way like it dies in a 1 month, and along with the death of the host there will be the death of the microorganism that is actually resulting into the disease. So it is not always like you have to modify the variety so that the plant could survive against the attack of the pathogens. No, another genetic engineering also includes the concept where you are programming the cell towards the death, so that along with the death of the host, there is the death of microorganism that is responsible for originating the disease, right? So the strategy of artificial programmed cell death, it has been designed, very important, so that it could mimic hypersensitive response. That means the responses which were otherwise to be going to be visible in 5 to 6 years, they are going to be quickly they are going to be quickly visible within the 1 month. And again I repeat, it will lead to the death of the host, and along with the death of the host there is going to be the death of microorganism. But how this programmed cell death can be induced? It is brought about by a gene action, and what gene action is endogenous. That means within the gene. That if there is a gene, then within the gene there is a certain promoter that will promote or that will lead to the quick cell death. And this endogenous gene action, this will come into the active state. Otherwise it, because it should not be like every time even if there is no attack of the cell and there is no attack of the disease, it is leading to the programmed cell death. No, that must not take place. Otherwise even without the attack of the disease, the cell may lead to the programmed cell death, right? So here endogenous gene action is there, that is actually leading to the programmed cell death, but it becomes active only when there is a particular stimuli, and that stimuli, presence of that particular stimuli will actually trigger activation of this endogenous gene action. And when it will activate, it will release certain say it will release a certain, say we can call it certain stimuli, and those stimuli will lead to the death of the cell. Then along with the death of the cell, there is going to be the death of microorganism that is actually present in the host, right? So this is a very important concept of genetic engineering. Here, cell death, artificially production of cell death, it is further divided into two systems. One is two component system and another is one component system. When we talk for the two component system, very important it is now. Listen carefully. Two component system. I will explain you along with the diagram so that it becomes easy for you all to understand. So this is the pathogen, right? And within the gene, this is okay. First, I explain you what is two component system. Here, two precisely matched transgenes, like two transgenes are there which matches with each other. Precisely means very accurate matched transgenes which are two in number. This is the reason it is said to be two component system, because here two transgenes are actually triggering artificially programmed cell death. One is this transgene, and this transgene is matched with this transgene that will produce a quick hyper sensitive response and will lead to the death of the host cell, right? This is clear. Two component system, that means here two transgenes are, this is cell, host cell. This is a pathogen that is responsible for causing disease to this host. But within this host, there is the presence of one endogenous gene action, and that endogenous gene action is involving the two transgenes. One transgene is AVR and other transgene is R gene, that in turn is producing CF9, right? So it is clear to all, like two precise matched transgenes are expressed in the same cell. This one transgene is AVR9, and this AVR9 in turn is, because any gene comes into action, because if genes are without any promoter, if they are coming into the action, they will lead to the death of even the normal cells. So any gene comes into the action if it has promoter for its activation. Like see, TV is not switched on. If you TV won't switched on until unless you switch on the like switch on, right? In the same way, this will not come in its into active state until unless it is regulated by the promoter, and this promoter will be regulated when pathogen will elicit, when pathogen will release the toxin, and this toxin will activate the promoter, and this promoter will in turn activate this transgene, right? So this is only what is written into the slide, like this. One of the transgene that is said to be AVR9, in turn it is regulated by promoter, and this promoter in turn is, when this promoter will be induced, like when it will be released, it will be induced by the non-specific eliciter, and this eliciter means something will, what we call eliciter? Eliciter means something that is जानबूझ के when someone teases some other person, like that is said to be liciter. Like itself it will tease this promoter, and after this promoter gets irritated, it will activate AVR9, and this action is in turn to the liciter, that is in turn is produced by the pathogen that is responsible for causing disease. So whenever this host is invaded, that means whenever this host is visited by this pathogen will, how it will be invaded by this pathogen directly is not entering into this host cell. It is actually producing the liciter. Eliciter means that is activating something, and this eliciter is actually aimed with the death of the host cell, but it does not know the programmed cell death, artificial program. It is not aware about the artificial programmed cell death, and whenever this eliciter is going to enter into this host cell, it is normally going to activate this promoter, that in turn will activate AVR9, right? That is one of the transgene. Now the other transgene is R, right? The other transgene is R, and this R is corresponding, because we said precisely matched transgenes. Precisely means very accurately two transgenes Because it is two component system, it should not be like the other transgene which we are looking after, it is no more working in accordance with this gene, then the total mechanism is going to fail. So the two transgenes which are to be selected, they must be one which are in correspondence, which are precisely matched with each other. That means when it get activated, it will lead to the activation of this gene, right? This is only the reason it is said to be two component system. Otherwise, the whole mechanism is going to fail, right? So other transgene here is not mentioned the R gene, but that R gene, this in turn will activate the R gene, and this R gene is produced in correspondence to this AVR gene. For example, this R, in this case, this is CFN9. This is the another transgene, right? And it will be driven by its, somewhere here will be present its promoter. Its activation will be the result of activation of its promoter. This is two component system. Now, here this is the process. This is the process that is actually taking place here. So what is happening in this two component system? Whenever this pathogen will attack this host cell, it will be attacking through the production of this non-specific elicitor. As a result, there will be expression of AVR gene, and this in turn is this is regulated. This will be regulated. It will be brought in the mode of action immediately following the pathogen attack. As soon as pathogen will attack, it will come into role, right? But only this is only in case of infected cells. Don't forget, this procedure is no more taking place because we are all about talking about artificially programmed cell death, and artificially programmed cell death is to be conducted only in the cells which are infected by the pathogen, right? So process is somehow goes like this. Here what is happening out, there is being the expression of AVR gene, and this AVR gene would produce the elicitor that is going to activate something else. And these elicitor would be recognized by this R gene. These elicitors are produced. These elicitors are going to be recognized by this R gene that in turn will activate CF9, right? And this will lead to this R gene is prepared in this way. This is the R gene, right? This elicitor is recognized by R gene. If it fits into it, this is the R gene receptor. This is the R gene, right? If it fits into it, that means it is easily recognized, right? And after the recognition, it will initiate and culminate in hypersensitive response. That means it will, the cell that was otherwise going to die in the 5 years, it will die in a few minutes or in few months or in the fewer weeks, right? This is said to be two component system. So this is what I have already explained to you all. This is two component system where this promoter is being active upon activation, it is being activated by release of the elicitor by the pathogen. It will release certain elicitor that in turn will be say these are recognized by R gene, and quickly they will produce the hypersensitive response. So here two components are precisely matched, and then they are playing a major role in programmed cell death that is artificially being produced. Now we will talk about one component system. So this is one of the thing that is to be noticed in case of say two component system. It is applicable to any pathogen combination, right? It is applicable to any plant pathogen combination, but the condition is AVR gene is to be driven by the appropriate promoter. That means this AVR promoter, this AVR gene is to be activated by a recognized promoter, right? I cannot say like if say this is promoter P, say there is another promoter that is written as this much. So it is to be activated by this only. It cannot get activated by this. So there must be, in short, I would like to say there must be the presence of particular promoter for the activation of the transgene, right? This is all about two component system. But in single component system, we do not require two precisely matched transgenes. Here one will be sufficient itself. And here transgene usable, this mechanism is like that is encoding. Here we are encoding the toxins. There what was being done, hypersensitive response was being given, quick response was being given, and that otherwise maybe the response may be given in many time. That response was given in a short quicker time. But here we are not looking after the hypersensitive reactions. Here whatever transgene that is introduced into the infected cell that is encoding for the toxin, say that in toxin is either any ribonuclease or other enzyme, and these toxins are going to be toxic to the plant cells. So here is the example. Here we are talking about Barnase gene. See, again pathogen will attack, but here what is being this endogenous system is producing toxin. We said na that toxin can be ribonuclease. Ribonuclease means the enzyme ribo. RNA is enzyme. Okay? That enzyme, this upon the attack of the pathogen, this is an infected cell. When this infected cell is attacked by the pathogen, it will attack the host by production of certain non-specific lysator. These lysator will activate the endogenous action that in turn is going to produce toxins, and these toxins will kill the plant cell. So along with the death of the plant cell, there is going to be the death of the this pathogen also. So here one of the example that could better explain one component system is here the Barnase gene from Bacillus amyloliquefaciens. It is placed under the control of infection, and the promoter in turn to it is PRP11. This was we are talking for this infection in terms of the crop, say potato, and the potatoes into which this transgene was introduced, they showed an effective control of this infestance disease, right? So this is all about one component system. So here this is the promoter. So here this is the promoter PRP11. It is specifying the expression of Barnase gene, and those cells which are infected by the fungal pathogen. This here is not mentioned here, but I would like to tell you, like whenever pathogen is attacking, endogenous system is resulting into the production of Barnase protein, right? Barnase gene is activated. See, very first this promoter is going to be activated. This will lead to the expression of this promoter will lead to the expression of this Barnase genes. This Barnase gene will produce the protein that is said to be RNase, that is toxin itself, and it will lead to the death of the cell, right? Which cell? Very important, the cell that is infected. We are not to artificially induce death of normal cells. Obviously, we need to lead to the death of the cells which are infected by fungal pathogen. As a result, pathogen will die along with the die host cell, and this is what we are calling it as artificially programmed cell death. So here in genetic engineering, till now what we have learned till now, whenever I talked for the genetic engineering, I said within the genetic engineering we always talked about the modification of the gene, but here gene is not being modified. Here system of the cell is being activated, so that because we know certain diseases, see students, there are basically two concepts. One parameter can be if any cell is affected by the disease, that disease can be controlled. But what to do for the other parameters where disease cannot be controlled? In this particular direction only, we need to look after artificial programmed cell death, right? So here we are activating the system of the cell itself, but the cell, that system will be activated only within the cell that is infected. Otherwise we need not to activate the cell, right? So that system is activated as a result of attack of elicitus, because we modify the gene in a way like we know whenever this particular eliciter is going to attack the host, it will always activate the promoter concerned with that particular transgene, and as a result it will lead to the death of the cell, and that is what is required in case we cannot control the disease, right? So this is all about genetic engineering. But one aspect is to be noticed in terms of genetic engineering also. So this is clear to all. Barnase gene is there that is producing Barnase protein, and that particular protein is RNase. Its production is activated by this promoter, and this is toxin in the nature, and it is leading to the death of the cell. So along with the death of the cell, there is going to be the death of this pathogen also, and one because since we could not control the disease, we can actually kill the, it is better to kill the cell where the disease cannot be controlled, right? So this is one component system. But here the strategy of artificial programmed cell will not be, it is not effective against facultative parasites, right? This is to be noticed. So this is all about today's lecture, where we have covered the breeding methods where we could create the plants which are resistant to the gene. So this was all from my side. If there lies any question, you can ask me right now, and even you can text your queries to me later on after the lecture also. So I'm putting you all. So thank you, and stay home and stay blessed.